So tonight I'm going to talk about building on the nanometer scale using DNA as a building block. And before I get into my talk, I actually want to do a quick kind of pre-talk calibration on two topics, both size or length scale and the motivation for working in this area. And I think we all have a lot of intellectual freedom, and we can pick what we want to work on. And I want to tell you why I'm really excited to work in this area. But first, just to calibrate our size, how big is a nanometer? It's actually very hard to imagine 1/1000th of a meter. You can imagine a meter, right? That's kind of a life length scale. People can be a meter, 2 meters tall, but 1/1000th of that is tough to imagine. Your fingernails grow about 50 nanometers a second. And so that's one factoid that I remember. Another way I think of this is if you look at the back of a US penny, if you ever look closely, you actually see there's a little tiny Abraham Lincoln in the middle of the Lincoln Memorial. And you can imagine little tiny Abraham Lincoln's eyelash is approximately 100 nanometers in diameter. So we're on the very small length scales here. And you can imagine trying to build on that length scale. Obviously, it's not going to be the traditional way that we build on the macro length scale. So that's really the size calibration. Next, I want to calibrate on the motivation for why do we want to build with biomolecules. And the way I think about this is really to do a quick thought experiment. And imagine that you were transported back in time to 1975 in the Bay Area. And say you got settled and found a job. What do you think you would do on your weekends? If you're anything like me, you would go find the Homebrew Computer Club and start attending those meetings. And then about a year later, you would see Steve Wozniak and Steve Jobs debut the Apple I computer. And knowing what you know now, back in 1975, it'd be quite obvious that this is a good group to hang around with, right? They're doing really exciting, innovative stuff. And it might not look like much back in 1975, but fast forward 38, 40 years, and now we have these kind of supercomputers in everybody's pocket with access to GPS and space and cloud computing, and there's all these amazing things. And if you can just kind of extrapolate forward, this is a really powerful general platform that we were building on back then, and then it's come so far. So for me, my kind of Apple I moment came in 2005 when I was a first-year graduate student, and I saw this image of DNA smiley faces. So this is atomic force micrograph, and I'm going to walk you through this in a moment. But the amazing thing to me is that this is 100 nanometers across, and that you could assemble any material into any type of shape that you wanted. In this case, a smiley face, but it really could be anything. That suggested that this is really a powerful technology. Even though it's not quite obvious what we're going to do with it at that time, it seems like a good area to focus on. Like this could have legs in the future. The other aspect is that we actually already know the answer of how powerful biomolecules are. And that's because bio-nanotechnology was really perfected by nature 3 billion years ago. And so we can look at the molecular machinery in our own cells, like here are a couple of example molecules like RNA polymerase, kinesin. The ribosome is maybe the most impressive molecule in the universe. And I can show you these movies or these animations basically of these molecular machines doing their job inside the cell. So RNA polymerase is transcribing RNA or DNA into RNA. We have molecular motors like kinesin that haul cargo to and from different regions of our cells. And of course, the nano compiler that life figured out 3 billion years ago is the ribosome, right? You're able to actually convert that RNA message into a protein shape that self-assembles and actually can perform biochemistry. And these are all nanoscale devices. And then, again, we can build with these materials. Nature's figured out how to build with these materials, and then you get amazing emergent behavior in these living systems, right? You get self-assembled construction, we all are self-assembled. We have adaptation in nature, healing, reproduction, even consciousness. So you can basically do anything with self-assembled biomolecules and plus a few billion years of evolution. So the way I think of it is like, how could you not want to build with biomolecules? Not like, why should we? But like, why would you work on anything else? That's the way I've thought about it. Okay, so that's our pre-talk calibration. Now I'm going to get into what we actually work on. And the first thing I want to tell you is that we use DNA, not in the traditional sense, so you might maybe think of it as an information storage molecule inside each of your cells. We actually think of it more like a Lego building block that we can use to build on this nanometer length scale. So why do we build with DNA? Well, it's actually relative to proteins or other materials. It's actually very simple, but it allows us to do programmable self-assembly. And I'll explain what that means later in the talk as well. We have to give credit to Ned Seaman, who's an NYU professor. He really pioneered this entire field, starting about 30 years ago. And he's still actively working in the field and doing amazing things, but he was really the visionary that saw DNA as our potential building material. So how do we build with DNA? Well, as I mentioned, it's this polymer. It's like a 1D kind of string, or like you could think of it like a piece of yarn or something. How do you go from a long string to create like basically from a one-dimensional building block to two-dimensional and then later three-dimensional? And the trick is really we can choose any DNA sequences that we want to synthesize. And so these two DNA molecules that I'm showing here, you could imagine linking them together by getting rid of the red and blue strand and actually resynthesizing them with slightly different arrangements of sequences such that they cross over. So you can see separate together. Now they're like spot welded together, and that's because we've encoded in the red and blue sequences this kind of junction, which is a holiday-like junction. And you can join neighboring helices of DNA in space using this kind of trick. And that was really what Ned Seaman pioneered, to make these little 4 nanometer by 7 nanometer little kind of building motifs. And so the next question we have is, okay, well, how do we get larger structures? This is quite impressive, but then we're meters in size, right? So how are we going to span those length scales? And this was really, there was a revolutionary breakthrough in our field called DNA origami that came out, as I mentioned, right about the time I was starting grad school. And so these are atomic force micrographs of a variety of different shapes, all built from DNA. And atomic force microscopy is kind of like a record player. You have a little tiny needle that scans over the surface and bumps up and down. And wherever it scans over some DNA, you can measure with a laser beam that's reflecting off of that some height differences. And so that information can be used to reconstruct these images. And so Paul Rodeman at Caltech, he invented this technique in order to build about 10 times bigger structures than anybody had really been building at the time. And so I'm going to tell you how this works by focusing on the famous, as Paul called it in the paper, disk with 3 holes, but it's actually recognizable as a smiley face. So the first thing you notice if you look at this is that we have hundreds of these crossovers, so it's actually much bigger than those smaller little tiles that I mentioned at the start. And how is this designed? Well, it's really made up of one long, so we say 7 kilobase, so 7,000 base long single-stranded DNA, which Paul called a scaffold. And that's self-assembled into the shape using about 200 short, we call oligonucleotides. So these are short pieces of DNA. So it's all made out of DNA, one long strand of DNA and a bunch of short strands of DNA. And we choose the sequences of those short strands to bind to the long strand using the base complementarity. And it forces everything to self-assemble into whatever shape we design. How does this assemble? I've been asked this question many times, so eventually I made an animation as a grad student. So I'll just show you my little animation here. It's not a simulation. It's just kind of to help you imagine that this is actually a self-assembly process where It's a one-pot reaction. You just mix everything together, heat everything up, and then gradually let it cool down to room temperature. And all of this, all the DNA, all has the information for the shape is encoded in the DNA. So all these strands find their places on the scaffold, and at the end of this process, you have the shape times billions and billions of copies. So it's really quite exciting and an interesting technology. And so that's where I came into the picture, right about the time we learned that Paul had developed the 2D version of origami. And so I worked with my graduate advisor, William Shih, to develop 3D origami. And so the idea here is that we can actually pack DNA on a 3D lattice. We don't have to make these kind of flat shapes. We can actually choose different crossover locations to get these multi-layered structures. And here I'm showing you negative stain electron micrographs. So this is just a different way of measuring or taking pictures of the DNA. And these are 20 nanometer scale bars. So you can see we're able to make a variety of different shapes with various cross sections. And really, we're just working out the baseline rules for self-assembly here. So we're not doing anything with it. We're just trying to figure out, like, what can you build? How do you build it? What are the conditions that gives you high yield of assembling these shapes? And so we published this method of making 3D shapes back in 2009, which feels like a long time ago. And so we can make lots of different shapes. We can choose kind of how they self-assemble. And then the question that everybody has at this point, after seeing the smiley face, after seeing this icosahedron, is usually almost the same exact question. What is this useful for? It looks like you're having a lot of fun, but when are we going to actually see a useful application? And I think there's definitely value to basic understanding of how things work, right? That's where the applications ultimately come from, is the initial basic questions that we're asking. You need to do that before we can get into the applied area. And so we've been working on the kind of basic questions, but we are trying to transition into real applications of this technology. And I really think that there are going to be many of them in the future. And I'm going to tell you about one that we've been working on recently. And we look at this technology and we think, well, could we build better cancer therapies using DNA nanotechnology? And so what we want to do is actually take inspiration from our own immune systems. So for example, cytotoxic T cells are able to actually compute whether or not a cell is healthy or infected with a virus. And once they make this decision of like, okay, I'm looking at an infected cell, they can actually send a message to that cell that you need to self-destruct. Something is wrong, time to shut down, stop infecting everybody else. And so they can trigger this programmed cell death or apoptosis in target cells and actually leave the healthy cells alone. So when you get sick, this plays out inside your body with, you might not feel that great, but it's not that bad compared to what we do with chemotherapy. So, which is a very non-targeted, or often very non-targeted type of approach for treating cancer. So what my colleagues and I asked was, could we start using DNA nanotechnology to kind of mimic this immune signaling and do this in a targeted fashion? So if you think about this, like this cell talking to an infected cell, you don't necessarily need a cell. It's just physics, right? It's just arrangements of matter at that cell surface. If we could recreate that synthetically, then potentially we could target cells, we could program their behavior and maybe even have them self-destruct. But then since we're doing this from scratch, maybe we could just do this targeted to whatever we want, including cancer cells. So the result of this is what we call a DNA nanorobot, which I'm going to tell you about tonight, and it actually is capable of targeting cells in the Petri dish, right, and delivering a payload. So this is using DNA origami to create what we call nanorobots. This is the initial design. It's a 35 nanometer DNA origami barrel, and it has inside these pink antibody fragments So that's really going to serve as the drug. It's going to bind to that cell surface, and I'll talk about that in a bit. This is a two-domain structure that is held together by these two hinges in the back and these kind of like twist tie latches in the front. And that I'll tell you about that in a moment as well. But first, let's look at this under the electron microscope. We can put gold nanoparticles inside. We can put antibody fragments. We can really program it like these to self-assemble and then put whatever we want. We kind of think of it like the space shuttle. It's a kind of general purpose carrier. The way the targeting works is, like I mentioned, this DNA lock. And this is a special sequence of DNA which, in the presence of a ligand or a key, and this could be a cancer marker or something on a healthy cell that we want to target, when that's present, it will actually bind to and displace the kind of double helix and unzip it. It will stabilize it in the kind of unzip state, and this is called a molecular beacon or a molecular switch. And so this had already been around for probably a decade when we repurposed it to actually control the open or closed state of our entire device. So we put a couple of those aptamer switches on the end of the barrel, and then when the ligand is present, it comes and unzips in those two locks, and then the entire barrel is able to swing open and expose its payload. And so these antibodies, which could be a very toxic drug that were sequestered, are now available to interact with the surrounding environment. And so here you can see the open version of this device. It looks very different. It's no longer kind of a box, right? It's splayed open. And again, we put gold nanoparticles in so you can see what it looks like when it's open. Okay, so it's one thing to build a model or a shape or something. It's another thing to actually build something that does something. So we actually have to do assays or tests in the lab to check to see, does this actually work as expected? The first test that we did is to test whether this is selective. So can we mix our nanorobots together in the same test tube with some target cells, which are shown on the far right. These NKL cells are natural killer leukemia cells. And so that's a cancer cell line. And we mix that together along with some other bystander cells. And so we want to see if we can target those. The way that we measure the targeting is we load fluorescent antibodies. So these are something we can measure where these nanorobots are going. And those antibodies will bind to both cell types if they're open. So if the nanorobot pops open, it'll bind to either cell type. And it's binding to a target called HLA, ABC. And then we use keys we basically program locks that will recognize only the so-called keys on the cancer cell surface. And that cancer marker is called platelet-derived growth factor. And the lock we used, we actually just took it out of another paper it's called 41T. So here's what we expect. Can we mix all these three together and then get targeted binding to just that cancer cell on the right and then just leave alone the other cells on the left? And so first we did this negative control. And this is with locks that do not recognize anything. That's just to give us the baseline. And so what you can see here, this is actual experimental data from a flow cytometry experiment. And it's this 2D plot where on the y-axis, we can tell the two cell types apart with a separate antibody. So you can see kind of our target cells on the bottom, our non-target cells on the top, are those little kind of clouds of little points there. And then the fluorescence of the antibodies is measured on the x-axis. So if this is on the left, those cells are naked. They're not bound by any of those nanorobots. And if they shift to the right, like in our positive control, so here we unlock the device. So first, negative, locked. You can see unlocked. Both types both of the peaks shift, right? So we are able to bind to both cell types if they're open. So what we're looking for in a selective nano device is the case where we'll see that shift on the bottom, but not on the top. And fortunately for our careers, that's exactly what we saw, which was a really great day in the lab. So I always tell my students that, science is very much about delayed gratification. So you'll do like months and months of work, and then you'll find out the answer of, did this thing actually pan out the way you hoped? And then if it doesn't, you just kind of do more test experiments and continue kind of at the same rate. If it does work, it's actually even more work to follow up because it suggests hundreds of follow-up studies. And so we were able to take a break for 5 minutes and celebrate, and then work really, really hard to do much more assays after that. So we did another assay to can we actually kill these cells by swapping out that cargo and delivering antibody therapeutics that will actually now bind to those cell surfaces and cause signaling that will induce cell death. And so we did a similar assay. Now, I'm not showing you the raw data. I'm just showing you a plot where we have increasing concentrations of our device, and we look at them the cells 3 days after one dose, and we can see that the negative control on the bottom basically is flat. The open or unlocked version, we can start seeing killing of those cells around the 1 to 10 nanomolar concentration, and then the selective version is similar. Once we get enough material around, we're able to do that as expected. So now what we're doing is basically, this was my postdoctoral work with Ido Bachelet in the George Church's lab. Having come to UCSF, we're continuing to work on scaling up the production, so we have to make a lot more of this material. And then we want to begin actually testing this in living organisms, cancer models in mice, for example, to see how do we design devices will eventually work in human patients. And I'll just show you a very quick update where for scaling up, we're actually growing our DNA materials in these benchtop bioreactors, so we can grow this in cells to very high density to get much more material than we typically do in shake flasks. So that's just kind of, we're continuing to this project and pushing forward. And so I think the early results are promising. We're going to continue working on this. And now, for the rest of my talk, I want to shift gears. And of course, we're at Autodesk, so of course I have to talk about CAD, computer-aided design. So hopefully after that, you're thinking, okay, well, how can I make one of these nanorobots? And it's actually much easier than you think, because in addition to doing all this wet lab work, I also spent a lot of time writing software as a student. And so I worked with a number of other students and staff during my graduate and postdoc years to develop this software called CAD Nano. And I'm going to just show you what it's like for us to design these structures by showing you how do we design a nanorobot in CAD Nano. So the first step is we route the scaffold in the software to create kind of the shape we want. So here's the interface of CAD Nano. And so you'll first notice that there's this kind of honeycomb lattice of circles in the top left. That's kind of our kind of drawing board. So we can add, by clicking on one of those circles, we can add to our data structure an empty helix. When we click on an empty helix, now we add a little piece of scaffold DNA to the structure. And if you click and drag everything together, it assumes you want to start by cross-linking those. And so we create a loop of DNA right off the bat. So you can already see the outline of our barrel is quite narrow. So we actually have to extrude it further. So we select all the edges and drag them out. And so now we have the barrel outline. And so you can see over on the right, that's the 3D model. So this actually runs inside Autodesk Maya. So we created a plugin in collaboration with Autodesk to help with our design process. And here I'm just adding some reinforcing strands of DNA around the edges. I don't want this to kind of smush down when we image it under the electron microscope. And so all the geometry of the DNA is just built into the software. So you don't really need to worry about like where you are in terms of the double helix, at what angle. You just click on these little icons that link together the DNA. And then we can rearrange the schematic just to make it a little bit more readable. But that's really how we first start thinking about making a shape, is just where do we want the scaffold to go at the end of the self-assembly process. Step 2, we create the staple strands. And so this is where we click on this stapler button, and it creates the full complementary set of DNA, including a bunch of crossovers. And so here I can highlight a few different strands, and you can see these are actually, we're not quite done with the staples. These are very long strands at this point. And so what we do is actually break those staple strands into smaller pieces as our next step. And we have some fun little kind of graph theory algorithms to break everything according to our parameters. And now you can see I've split those long strands into much shorter strands, 40 to 50 bases in length. And now we could actually export those sequences and order them right now. But of course, we're not done yet. We actually want to decorate our structure with functional elements. Excuse me. So here, this is where the 3D interface is actually really useful. So we can actually see every base position of all the staples on the right, and we can just really navigate in 3D and then edit in the 2D interface and start making attachment sites for our antibody fragments. And so hopefully you get an idea. So here's what I'm doing. I'm attaching that red strand. I'm going to route it and then create a crossover so you have a little strand hanging off right at that position. And that I'm going to add the complementary strand linked to the antibody later, and that will dock it to that site. And then I can fly 15 nanometers away and add another attachment point. If you want to start thinking about decorating the outside of the structure, say to improve circulation in the bloodstream or something, then you could just fly out there and add another attachment site. So hopefully this gives you an idea that really we can start designing and like in a matter of hours create these shapes using the CAD software. Step 4 is the sequence assignment. So here I've already added in the extra attachment sites for everything, and then the lock sites for those aptamer sequences. And so what we're going to do is now zoom in and actually assign the actual A's, T's, C's, and G base pair sequences. We route the scaffold, and then the complementary base is assigned to every staple. And now we can actually just export those sequences, and this is really the output of the program, is a list of DNA sequences. And so this is very different from normal CAD, but this is the output. And we can just send that to our oligosynthesis company, and then the last step is to actually assemble these. So we get each strand of DNA in a different well, the 96 well plates. We combine all of those together, mix them with the scaffold and some buffers, and put them on the temperature control block, and then heat everything up like I showed in the early animation, and then cool it back down, and then at the end of a few days protocol, you get 50 billion copies of your device in about the size of a raindrop. So, and this is a really fun process, and it takes, actually one of my students is here, and when she joined the lab, she had no experience with any of this, and she actually had EM data by the end of her 3rd week in the lab. So, the software and the methods are worked out to where we can really rapidly prototype new ideas. And so I just want to close on some topics that are actually very near and dear to my heart, which are really not just doing the research in the lab, but also sharing these ideas with the community and also kind of building this field from the bottom up. And so one thing I think about a lot is like, how can we recruit and educate students? And I was really inspired as a graduate student working as a TA for the iGEM competition. This is the International Genetically Engineered Machines competition that's held every year at MIT. And this is really about engineering bacteria, and this is in the field of synthetic biology. And I looked at this competition and thought, wow, this is amazing to see how inspired all these students are to learn these methods and build new things. I would love to have something like that for our kind of nanoscale design field as well. And so when I was a postdoc, I went and asked the director of the Wyss Institute, could I have some money to start my own iGEM? And amazingly, they said, yeah, sure, why not? Just write a proposal, and then I did. And actually, we ended up starting Biomod, which has now gone for 4 years. And the first year, we had over 100 students from 10 different countries. So here's our group photo. Second year, we had about 150 participants. This actually just took place, the 4th year just took place this last weekend. So I was in Boston, that's why I have a cold coming back. But I'm going to show you a quick clip of just some highlights of the competition. And so these students, they work over the summer to design things, like shapes or devices out of using DNA nanotechnology or RNA or other biomaterials. And then they make presentations and then they present their work along with a project website, and they actually also have to make a YouTube video explaining in plain English what are they doing and why, what does it mean? And they actually act out skits and do these amazing presentations. This person, they're quite dramatic. This was my favorite. This guy's showing fluorescence resonance energy transfer, and they were dressed as fluorophores. And so this has exceeded all of my expectations so far. So these are the students. This was just on Saturday and Sunday. There's our group photo. And so these students are every year doing really amazing things and actually contributing meaningfully to our field, and hopefully will, in the future, continue to work on this stuff. So if you want to check this out, this is at biomod.net. Another thing I think a lot about is how can scientists communicate about our research to people that don't necessarily have years of training. And I think social media and animation are a great way to do that. And so when I saw this whiteboard animation format, I was really excited to actually make my own whiteboard animation along with my friend since first grade, Jason Brown, who did the drawings, and I narrated. And basically, this is just explaining kind of what is bio-nanotechnology. Do you know that we can actually just order DNA to be synthesized from a company and they'll ship you the DNA? Like most people don't work with this, so you wouldn't know that. But you can just order for $10 a piece of DNA and they'll ship you a tube 2 days later. It's amazing. And so we have close to 45,000 views now. And so this is, I think, I can sleep and people are still learning about this stuff, which is great. And then the last thing is, building a nice lab website. I also collaborated with Jason to just make artwork. And if I spend 100% of my time working on this stuff, that leaves, you can do the math, 0% of my time for hobbies, unless I figure out ways to integrate them. And so I try to figure out ways to take things like art or animation or building websites and incorporate that into our research. So I still get a chance to do stuff like that. And so the point of this website is really to trick graduate students into thinking it would be fun to work in my lab. And so they come in and they, build some nanostructures, and then they find out, what it's really like. And we're in Mission Bay, the new UCSF campus. And if you want to learn more, our website is at bionano.ucsf.edu. If anybody wants to volunteer or has programming skills or wants to contribute some way to the lab, I'd love to hear from you. And with that, I'm going to just acknowledge all the people that made this possible. It's not me by myself by any stretch. So my group members, and many of which are here, and UCSF, of course, for giving me my one job offer during this interview process a couple of years ago. I'm really thrilled and couldn't imagine being at a better place. Funding agencies, Autodesk has actually been the lead supporter in addition to the Wyss Institute for Biomod, so they really make it possible every year by contributing to help cover some of the costs to run the competition, and that's been really fantastic. So, and then of course my PhD and postdoctoral advisors and colleagues, and with that, let me just thank you for your attention. I'd be happy to take any questions. We have time for, I'm told, 2 questions. First question right here, and then.
How many strands is it to create that little cage with the trigger mechanism? And how do you get the antibodies and the ligands?
Okay, so the design itself is one long scaffold strand, and then about 175 short oligostrands. But then, of course, you need billions of each in order to do the self-assembly. The way that we get the antibodies and the ligands you can actually order all of this stuff from companies, so they'll synthesize them and send them to you, or we can actually grow cells that are expressing those materials. And so, for example, a cell has the ligand that it's expressing, we just grow those cells and they're making it, and so we mix the two together, and they'll find each other. Yeah, right up front.
So with using DNA as your building material, is there any sense whether putting it into animal models or even humans that it will be immune privilege? What about immune response? And is there any track record of other people doing this?
Yeah, exactly. So that's a great question. So if we design all these exotic, elaborate devices, and then we put them into humans, and then we find that you generate a really strong immune response, how are we going to overcome that? And I think there's a number of different threads to this. I think I'll say two things. One, what we can do with DNA, it's highly programmable. So if there's a way around this, we have many options to try. We could decorate the structures with different peptides or proteins, or encapsulate them in things that will maybe mask them from the immune system. So that's one area. The other area is that we don't necessarily lose if we're generating a very strong immune response. We could actually kind of flip that around and say, well, can we actually recruit the immune cells to perform specific tasks? And part of that could really be generating a strong immune response and then controlling it. So even if we build devices that end up being very immunogenic and we can't overcome that, we could still make more powerful and more useful immune technologies and sort of leverage that as a positive aspect as well. So, but the programmability sort of helps you achieve either goal, and my hope is. But yeah, we have to do the experiments to find out. So. I'd be happy to talk to anybody afterwards if you have more questions. So, but I guess back to the party. So thank you very much.